Today I'm presenting ISMB ECCB poster 721 titled Automating Structure-Based Design, Integrating Fragment Hotspot Maps in Drug Discovery Pipelines at Scale. Hi everyone, I'm McGreig, a structural bioinformatics research scientist at Exscientia. Structure-based drug design is a powerful approach to discover and optimize novel therapeutics and small molecule probes. However, significant challenges still exist in scaling up analysis to include all relevant protein structures available for a given protein target, as well as in efficiently using the resulting data. The work presented here outlines two example workflows developed at Exscientia to automate several aspects of structure-based drug design: target assessment and de novo ligand generation. The workflows complement each other, both incorporating multiple successful methodologies such as fragment hotspot maps and leveraging cloud-based infrastructure to support the development of novel, precision-engineered drugs. We've applied our automated workflows to AlphaFold 2 models of the kinome and are presenting the experimental data validation here. The workflow begins with tractability assessments for each target. We first capture data from all available protein structures. For these structures to be of use, they must be prepared for druggability assessments and automated design. Once prepared, the structures undergo fragment hotspot mapping to identify cavities and contributions of residues to binding. Cavities for all structures are then clustered into pockets, which enables progression from a target-level method into a more detailed pocket-level analysis. In Figure 2, we see cavities identified across all publicly available structures for a given protein target using hotspots. The example here is PL-Pro. The pocket clusters are defined based on the overlap between the uniprot accessions and residue numbering of each cavity. This enables pockets in similar space to be combined, whilst retaining data around the conformational differences present in different structures, as well as capturing how frequently a pocket is present for a target. In addition, the score distributions across a pocket are utilized for comparison to a benchmarking dataset as shown in Figure 1. Here, the distribution of each pocket cavity to a benchmarking dataset of druggable and less druggable proteins enables the characterization of pocket tractability. A score of 14 or more indicates that a target is tractable, with values 12 to 14 as possibly tractable and less than 12 as likely not tractable. Residue-level features are also captured for each site residue. In viral targets, such as PL-Pro shown here, conservation metrics including amino acid frequency, Jensen-Shannon divergence, and feature frequency are examined across related viral species. In human targets, amino acids which differ to aligned off-targets are identified. The next steps of the workflow involve utilizing structure-based drug design-ready structures and automating the design of compounds for a given target. We applied this method to kinome AlphaFold 2 structures to showcase the capabilities of the workflow and to also identify the usefulness of homology models of design. We hypothesized that if even the lowest quality models are usable, then there is value running the workflow for the whole kinome. We therefore selected kinase targets that had no representation in the PDB, here selecting DIRC1B and PKD1, with assays available and at a range of AlphaFold 2 model confidence levels. The kinase domain of the AlphaFold models was extracted and the compound matter identified for these targets. A pool of ligands from the Cliffs ligand dataset containing compounds from all kinases aligned in a common reference frame was used as a starting point for the design process. Hotspot maps for the structural models were used to trim back the ligands to just their key pharmacophores. 50 fragments were then selected, balancing diversity and hotspot complementarity. Our generative design method, Gambit, is then used to search the chemical space for molecules that score highly under the reward function selected. The algorithm involves a starting population of molecules by applying operators inspired by natural selection. 50 parallel Gambit runs are performed for the 50 fragments, designed to reward synthetic accessibility, drug likeness, potency, RMSD to the original fragment position, and hotspot complementarity. The output compounds are scored, filtered, and pooled from each parallel run to provide the top 50 compounds selected based on diversity and properties. For this proof of concept work, for each target 9 compounds were selected from the 50 provided by Gambit for synthesis. For both DIRC1B and PKD1, no compounds are observed in Chembl, highlighting the novelty of the compounds produced. For each target, 4 out of the 9 synthesized compounds were active. Despite having a less confident AlphaFold model, With less than 40% similarity to known structures, our workflow designed micro and nanomolar hits against PKD1, opening the possibility for large-scale kinase design. To conclude, our automated cloud-based workflows provide large-scale structural analysis and yield valuable data and insights for target evaluation and design cycles. The multi-scale data aggregation provides both a top-level overview as well as detailed pocket information capturing structural outliers and insights into validity and risk factors for each site on a target. We have demonstrated the validity of the workflow on kinase homology models with active compounds designed and synthesized for each target. The modular nature of our workflow enables each stage of the current baseline to be replaced by future superior methods. For example, we've now computed fragment hotspot maps PDB-wide for both monomers and complexes, as well as on all human AlphaFold2 models. This analysis also considers all structures for a given target, which can now be incorporated into our workflow, finding the optimal structures for design and highlighting the conformational differences for a target. In order for our workflow to run across the proteome, compounds from outside the kinome are needed. This dataset can be generated on a per-target basis, using in-house tools to incorporate ligands from structurally related proteins, creating a larger compound pool to draw from for chemical starting points. The reward function can also be updated to incorporate molecules targeted towards conserved positions or specificity determining positions to generate better molecules for a given target. Overall, our validated end-to-end workflow highlights the power of automation, with innovations and future improvements making proteome-wide designs a future possibility. Thanks for listening. Any questions, please contact me at the email address shown.